In this video, I will show you how an entry in viruSITE database looks like. I will show you two types of entries, one for virus and the other one for a protein. So let's say I want to get some information about this herpes virus, and here is the entry for human herpes virus 7. Here on the top are located three quick links. One is to genome browser. I will tell you about this genome browser in a separate video. Then link to genome sequence. And the third one is link to relative genome sequences. You can see that 9 genomes are relative to human herpes virus 7. Relatedness among genome sequences is inferred from BLAST results. Here you can see e-values and also alignments, sequence alignments from BLAST. When I click here, schematic visualization of sequence similarity will be displayed. Circoletto and Circos applications were employed for this job. Okay, so now I'm back in the entry for human herpes virus 7. In this division, you can see taxonomy of this virus. Moving mouse over this plus icon, alternative names for this virus are displayed. And clicking on these icons here, all viruses belonging to this taxonomy group are searched in our database. So, for instance, here are all herpes viruses, all viruses belonging to this family, and also all genes, all proteins, and all RNAs. Here on the right, you can see the host organism for this virus, and again also alternative names. In this division are listed features of this genome. So it is genome type, genome length, GC content in the genome, percentage of coding sequences, and number of genes, proteins, and RNAs in this genome. Here, as you can see, are links to other online databases, RefSeq database with the reference genome sequence, links to GeneBank, to taxonomy database, to viral zone database, and so on. So, for instance, I click here and I am redirected to viral-zone database and to herpes viridae section of this nice database. And finally, on the bottom are located references to scientific papers and links to PubMed database. Okay, now let's see some protein entry in our database. So, for instance, this envelope glycoprotein. Again, on the top are quick links to genome browser, but in this case the display, the visualization is focused on this particular gene in the genome. The linked sequence contains amino acid sequence of this protein, and here is a link to search for homologous proteins. And you can see that 3 proteins are homologous to this envelope glycoprotein. Here there are. Again, sequence homology was inferred from BLAST results. You can see e-values and sequence alignments. Now I can proceed to description of this protein. You can see names of these proteins, position of a corresponding gene in the genome, length of the coding sequence and of the amino acid sequence, molecular weight of this protein, and isoelectric point. Some notes. Here is the gene that is coding this protein and also the source virus. Here on the right are numerous links to entries for this gene in other online databases. So it's NCBI gene database, ENA NCBI protein, UniProt database. You can see also gene ontology terms associated with this protein and PFAM and Interpro domains. When I click on this icon, all proteins possessing this protein domain are searched in our database. And you can see that 11 proteins from 11 viruses do carry this specific protein domain. And on the bottom are located references to scientific papers and also link to PubMed.